Because bacteria are so small, people often forget they are three-dimensional objects. Our protocol allows the accurate reconstruction of the shapes of both flat and curved cells. This protocol only requires slight modifications to a standard microscope and the readily available MATLAB scripts.
Before beginning the experiment, place 2 stacks of each of three 20 by 20 millimeter cover slips on the opposite ends of a single glass microscope slide. Pipette 200 microliters of agarose pad solution onto the slide, and immediately and firmly place a second slide onto the stack of cover slips to flatten the agarose. After allowing the agarose to solidify for about 1 minute at room temperature, carefully slide off the top slide and use a large end of a 200 microliter pipette tip to cut out individual 5 millimeter diameter pads from the gel. When all of the pads have been obtained, pipette the E. coli cell culture multiple times to disrupt any cell clumps and to ensure that the cells are homogeneously distributed, before adding 1 microliter of subculture onto each pad. After letting the samples air dry for 5 to 10 minutes, confirm that the droplets have been completely absorbed into the pads, before placing a cover slip onto the pads. Then, use a cotton swab to gently brush an appropriate sealant around the edge of the cover slip, taking care to keep the sealant away from the top of the cover slip. Immediately after sealing the cover slip, place the slide onto a microscope stage, and, after 5 minutes of thermal equilibration, use the microscope focus wheels to bring the middle of a cell into focus. In the associated